Good morning, everyone. This is Rebecca Bailey, and I will be your moderator for today's June webinar. I want to let everyone know that the speaker today will be taking questions at the end of the webinar, and you can ask questions at any time during the webinar by entering the questions into the questions box on your GoToWebinar side panel. Our speaker today is Dr. Jared Weddell. I'd like everyone to give a very warm welcome to Dr. Weddell. Jared is a senior manager in clinical pharmacology and exploratory development at Astellas. He received his Ph.D. in bioengineering from the University of Illinois at Urbana-Champaign, focusing on computational and systems biology. Since then, he's worked in clinical pharmacology and pharmacometrics, primarily supporting model-informed drug development across different therapeutic areas, including oncology, neurology, and rare diseases. And we're welcoming Dr. Weddell today. I wanted to mention that his paper was posted on our Twitter feed and was widely thrilled to see. And so with that, I'd like to give a very warm welcome. Welcome, Jared, and we're looking forward to the webinar.
Great, thank you, Rebecca, and thank you to Rosa for letting me come and speak with you all today to share some of this work. So hello everyone, good morning, good evening, good night. Today I'll be talking to you about QSP modeling to help us understand cytokine dynamics clinically for bispecifics that are dosed in solid tumors. So just to kind of help orient us with what we're talking about and what we're going to be focusing on today, I'll give a little bit of a background on bispecifics and the problems we see with them. I think everybody is probably familiar with bispecifics, but this is a very promising class of therapeutics, especially for oncology, but also across a couple of different therapeutic areas. Today I'll really be focused on oncology, and I really want to orient us for the sake of this webinar that when I talk about bispecifics, there's many different kinds of bispecifics. I'm really going to be focusing today on these T-cell engaging bispecifics. So we've got a bispecific where one arm binds to a T-cell, and then again, since this is oncology we're focused on today, the other arm of your bispecific binds to some tumor-associated antigen, and it leads to our process of helping the T cells selectively kill our target tumor cells, which leads to cancer cell death, things like T cell proliferation and release of different granzymes, perforins, different cytokines, chemokines, etc. So again, really for the sake of today's webinar, I'm really focused on T cell engaging bispecifics for oncology, particularly CD3-based bispecifics. So bispecifics that bind to CD3 with one arm on the T cells, and then some tumor-associated antigen with the other arm. And recently we've seen, bispecifics have really gained a lot of traction in drug development. There's been a lot of interest in applying these bispecifics to try to treat different indications. This is one example I threw up on here to kind of help orient with what we're kind of seeing with bispecifics currently. So there are some bispecifics, particularly Tecvayli, that was recently approved for fifth-line RR multiple myeloma. And just to kind of give you an idea of why we think bispecifics are such a promising class of therapeutics, I'm showing here some percent ORRs we've seen from registrational trials of BCMA targeting therapies. So these are, again, 4 therapies, the bispecific Tecvaylis at the bottom, that are approved for fifth-line RR multiple myeloma. So you can see Blenrep was an ADC that was approved with about 31% ORR. We've got 2 CAR-Ts, Abecma and Carvykti, that were approved that have about 73 to 98% ORR. And then here we've got our CD3 or our bispecific Tecvali, that's got about 68% ORR. So I think that what we're seeing with bispecifics is we're seeing a very nice balance between efficacy and kind of ease of dosing. You can see that this 68% is a lot higher than we get with the ADC. And while it is less than what we're seeing with some of these fifth-line CAR T approved therapies, these CAR T therapies are, I would say, more complicated to actually administer, and they're a little more complicated logistically with how we give them. These bispecifics, they're set up to be a little bit more, I guess, ease of dosing. We kind of have our standard sorts of dosing approaches with them, whereas CAR Ts, these can get pretty expensive. We have to get into a little bit more precision medicine with how do we extract these CAR Ts, how do we engineer them, how do we dose them in the patients. And so I think this shows that there's really sort of this nice balance of bispecifics of efficacy and usability that really makes them this sort of promising class of therapeutics. But one thing that we see with bispecifics that can lead to adverse events is this cytokine release. So again, I'm really focused here on CD3-based bispecifics targeting tumor cells, but we see that these cytokines are released as a result of this mechanism of action of these bispecifics pretty much in every single case. You're going to have cytokines being released from all of these mechanisms of T cell proliferation, through the mechanisms of how the T cell kills the tumor cells, and then once the tumor cells are dying themselves, they're going to be releasing different things that will then go away from the site of action, undergo organ disposition, and once these cytokines start to undergo organ disposition, they can lead to some nasty side effects, particularly cytokine release syndrome, or CRS, as I'll be referring to it through the rest of the webinar. And we've seen, across these bispecific therapies that have been studied clinically, CRS is one of, if not the leading dose-limiting toxicity for these bispecific therapies. Now, CRS can manifest in a lot of different ways. This schematic on the left shows a lot of different ways that CRS can manifest depending on which organs are kind of being attacked by the released cytokines. And the symptoms of CRS can kind of range from relatively mild to life-threatening. So if anyone's familiar with CRS, I'm not going to get into the nitty-gritty of CRS, but it's typically graded from grade 1 to grade 4, where grade 1 is sort of the least severe and grade 4 is the most severe. And generally, with grade 1, grade 2 CRS, these are things that would be like headaches, fevers, fatigue, things that are definitely uncomfortable and undesired for patients, but generally not anything that requires halting our drug, starting secondary therapies to help mitigate CRS or hospitalization. But once CRS becomes severe enough, particularly grade 3, grade 4, that's where we start getting into life-threatening sorts of side effects, things like organ failure, heart failure, respiratory failure. These are patients that at this point could require hospitalization, they could require intubation and or ventilation. And we really want to make sure, as we're thinking about these bispecific therapies, that we kind of understand what is our potential for CRS in these patients. Because we want to make sure that we're not getting to those potentially life-threatening grades of CRS. And again, I kind of mentioned this, but in terms of clinical management, there's a couple of different ways that we can manage CRS once we start to see it within a clinical trial where we're exploring a bispecific. So there's the scenarios where we might have to stop or de-escalate our bispecific therapy. There's different CRS mitigating therapies such as tocilizumab or siltuximab that we could start to give. Or like I mentioned, if it's severe enough, it might require hospitalization. So obviously these are all things that we want to avoid, especially during first in human or sort of exploratory trials with these bispecifics, as this can really result in not being able to accurately find a dose that's safe and efficacious for our patients. And this slide is just to kind of highlight that, dose selection, I think, for bispecifics is complex, but it's also very critical, not just from a cytokine release perspective, but with bispecifics, we also see some additional mechanisms that kind of lead to this dose selection being complicated. So one effect that we see with bispecifics is it's called a lot of different things. One name for it is this prozone effect. It's also called hook effect, the inverse U-shaped bell curve. But there's an idea with these bispecific therapies that as you increase your dose, you're not going to just see some increasing efficacy that eventually plateaus. With bispecific therapies, we expect that as you increase your dose, you're actually going to see an increase of efficacy up to some peak efficacy. And as you continue to increase your dose beyond that, you're going to start to see decreased efficacy. And the reason for this is because, again, your bispecific is binding two different targets, and the goal is to serve a bridge between these two different targets. If you go too high of a dose, you can independently saturate your two targets and not form a bridge between the two targets. So that's why it's really critical as we're thinking about dose selection for bispecifics, not only to think about the CRS safety perspective, but to also think about this efficacy perspective. How do we ensure that we can kind of pick a dose that is high enough on this efficacy curve that we see efficacy, but not going too high that we're over saturating our two targets independently and not leading to efficacy. And then in terms of toxicity, depending on what we think might occur with toxicity for our bispecific, we might expect to see this kind of inverse U shape with toxicity, but we might not. I kind of drew it here as, theoretically, maybe we'll see as you increase the dose, there's some peak toxicity beyond which you'll see a decrease in toxicity. I think that would occur if we believe that it's really the site of action, the tumor, that leads to toxicity. But it's also possible that, as you're increasing your dose, you're just going to continue to see toxicity increasing, as you can still have toxicity resulting from these two independent targets being bound. So this prozone effect, again, it adds this complexity to our bispecifics where we have to think about how we dose it, but it's really critical to kind of keep that in mind. And then there's another aspect of bispecific dosing that's gained a lot of traction recently that further kind of complicates how we think about dose selection, and that's this idea of step-up dosing. So again, this is looking at it from a safety perspective, but the idea here is if we, rather than just giving a flat dose regimen, so let's say our target efficacious dose for bispecific, this is just theoretical data, let's say our target efficacious dose is 50 milligrams once weekly. Well, if we just give this 50 milligrams once weekly flat, starting at time 0, maybe we'll see something like a 88% CRS incidence. The idea with step-up dosing is rather than going straight to our efficacious dose level at day 0, we're going to give lower doses and step up to that efficacious dose. So for example, let's say we want to give one step-up dose of 25 milligrams, and then the next week, we'll start giving our target efficacious dose of 50 milligrams once weekly. Or we could do two step-ups, where we're going to start with 10, step up to 25 a week later, and then step up to our target efficacious dose of 50. The idea here is that by doing this step-up dosing, we are reducing the total CRS incidence that we're going to see within our patients. And that's going to, obviously provide a better patient experience. We're going to have less side effects for these patients, and we'll also see less dose-limiting toxicities within our studies. So this slide is just to kind of highlight, I think that obviously for every drug that we study, dose selection is extremely critical. But I think when we're talking about bispecifics, there's these kind of two added complications of prozone effect and how we want to think about step-up dosing to mitigate our CRS incidence that kind of make dose selection for bispecifics maybe even a little bit more complex than something like, a monoclonal antibody therapy, for example. And so looking at some of the approved CD3-based bispecifics that are currently on the market, this slide highlights 4 of them that are currently approved. And there's a lot of information on this slide, but the 2 things that I really want to get across are, first, that all 4 of these approved CD3-based bispecifics have pretty complicated dose regimens, and none of them look similar to one another. And secondly, all of these CD3-based bispecifics have seen CRS incidents across their clinical trials. So let me just pick on a couple just to give you an idea of what this looks like. So blinatumomab, for example, this is a CD3 bispecific for treating RR acute lymphocytic leukemia. So this drug is given through basically a continuous infusion pump over 28 days, and then there's a period where you are treatment free. So this blinatumomab bispecific has a completely different dose regimen than the other 3 on this slide. This is the only one that's given sort of continuous infusion followed by a non-treatment window. And we do see that while it has the lowest CRS incidence across these 4 compounds, it still has CRS. There are still patients that are experiencing this. Versus these other 3 bispecifics, they all take this sort of step up dose regimen approach where they're starting at a lower dose and then they're stepping up to their target efficacious dose. In the case of mosunetuzumab, they're actually stepping up higher to 60 milligrams, and then at day 42, they're going lower to a 30 milligram once weekly dose regimen. So this is, again, I'm not going to go to all of the details of this slide, but this is to just highlight that these approved CD3-based bispecifics out there, they all have these pretty complicated and very different from one another dose regimens to try to mitigate that CRS incidence and to make sure that we can balance that safety and that toxicity. So I think that leads into kind of our main question of the day, and that is, how do we select a first-in-human dose regimen for a bispecific? We've got a new bispecific, it's entering the clinic for the first time. How do we think about selecting our dose, particularly that step-up dose regimen, so that we can balance this safety and this efficacy? And so if you approach it from the perspective of sort of efficacy independently or safety independently, this is how we can kind of think about it. So on the left, when we're thinking about how to select a first-in-human dose regimen for bispecific in terms of efficacy, there's really not anything too different that we'd kind of do here compared to sort of our normal model-informed drug development sorts of approaches. Typically, we'll be running our preclinical studies where we'll be collecting something like, here's our mice xenograft data, looking at tumor size across different doses of our bispecific to get an idea of the potential for this bispecific to reduce tumor growth. And then we'll have something like a mouse or cyno PK study to help us get an idea of what the PK would look like. And using the combination of the preclinical PK and the preclinical Ph.D, we can do sort of our traditional translational approaches to predict the human first-in-human dose from an efficacy perspective, which might be based on MABEL in this case. And I'll highlight that all the data in these slides is synthetic data. So this is just to kind of give you an idea of how we typically be doing this. So again, from the efficacy side, not really anything too different than how we would normally approach selecting a first-in-human dose. But I think once we start to really think about this from a safety perspective, this is where I think that things get a little bit trickier, because we can run preclinical safety studies to help us understand what is our potential safety risk once we enter the clinic. We can do things like in vitro studies, looking at, for example, release of different cytokines from different cell types across different doses of our bispecific. Or we could do mouse or cyno tox studies where we look at different doses of our bispecific across time, what are different systemic cytokine concentrations that were released from a cyno study, for example. But unfortunately, these preclinical studies from a safety perspective are usually not readily translatable to human. Because we know that, for example, whenever we do these sort of cyno-tox studies, these are usually healthy cynos. They don't have the tumor that our patients will have. We just know that there's so much going on in terms of differences between the tumor and microenvironment in mice and human, differences in the immune system, that it's really hard to kind of take this preclinical safety tox data, particularly cytokine concentrations, and readily translate it to human. And so that kind of leads to the goal that I'll be hopefully trying to work towards for the rest of this webinar, which is, can we predict what we expect clinical cytokine dynamics to look like before entering the clinic to help us support dose selection, particularly first in human dose selection for these bispecifics. And so to try to meet that goal, I'm going to turn to QSP, quantitative systems pharmacology. So I know for this audience, I don't need to cover, what is QSP, but I always like to just include a little brief background in case, anybody who's maybe doesn't do it hands-on is listening, or just to make sure that for those of us that might have a little bit of a different definition of what QSP is, that we're all on the same page for what exactly I'm talking about. So for me, when I think about QSP, this is a little bit of a wordy definition, but the way that I like to think about it is, it's these mechanistic representations of pharmacology that try to capture relevant disease pathology, drug disposition, and drug disease interactions to give this holistic, quantitative understanding of both the disease biology and the anticipated treatment effects. And so why is it that I'm going to be turning to QSP to try to meet this goal of predicting our clinical cytokine dynamics? Well, there's kind of two reasons, two nice features of QSP that I think are really good for meeting this goal. So the first is that when you're talking about QSP modeling, you actually do not need to have the data of interest in order to build and use these models. So again, this is a area that I'm not going to dive too deep into, but when we talk about QSP modeling, a lot of the time we're talking about these models that are built on first principles. So you can build in equations, you can build in parameters, you can give inputs based on first principles of biology. We know that this target binds to this target with this sort of mass action kinetics. We're going to parameterize it with these parameters that we kind of know from preclinical studies, in vitro studies, that we can estimate in different ways. And the nice thing I think about QSP that a lot of other modeling approaches really can't offer is this idea that I want to be able to model my cytokine dynamics over time. I don't need data of cytokine dynamics over time to be able to model that. I wouldn't be able to do this sort of modeling approach with exposure response analysis, for example, because again, this drug has not been studied clinically. I don't have exposures, I don't have responses, so I can't do that type of modeling approach. Hence, we'll turn to QSP. And a second nice feature of QSP, which I'll admit is not unique to QSP, other modeling approaches do this also, but still a nice feature for why we're going to turn to QSP is the ability to translate similar data. So what I mean by that is, again, we don't necessarily need the data that we're specifically interested in order to build these models. We can build it with basically no data from first principles. But if there is data out there that's similar to what we want, for example, let's say there's a competitor that has some data, maybe they're a little bit further in clinical development than us, they've started first in human trials and they've published some data. Well, we can start to take some of their data and leveraging it to help understand and predict what we expect our data to look like. And whenever I start these kind of new QSP projects and I'm interested in building or developing QSP models, I think it's always kind of a good first step and a good practice to see what is currently out there. Is there anything that's been published? Is there anything that's sort of readily available that you can take and kind of use for your own program goals and your own purposes? And so for this goal, again, our goal is to predict the clinical peripheral cytokine dynamics from bispecific dosing in solid tumors. From a search of what's out there in the literature, there are some great QSP models that are currently out there that look at bispecifics and even some QSP models out there that look at cytokine dynamics following bispecifics. But unfortunately, there's really nothing out there at the time that I started this work that was looking at bispecifics, particularly in solid tumors. So for example, this publication on the left, this is a fantastic QSP model that looks at how cytokines are released from blinatumomab and mosunetuzumab. But again, these are lymphomas, sorry. So these are lymphoma targeting bispecifics, not solid targeting bispecifics. So this model is not really, super readily available to be used to meet my goal. I would still need to kind of take this model, translate it over to solid tumors, figure out how to do all that translation. And then on the right, there's some other models out there that have looked at different approaches for, understanding cytokine release from bispecifics. But again, these models kind of really couldn't be readily taken and used because while this model in the bottom did kind of look at solid tumors, it wasn't really done in a way that could readily be translated to other compounds of interest. It's kind of more of an empirical model that wasn't readily translatable for our purposes. And so the survey of literature revealed that while there are some great QSP models out there, there's nothing that's really readily available to meet my goal. So what I was looking around for instead was finding out that there might not be any QSP models that can readily meet my goal, but there is some clinical data out there that is publicly available that I can use to help through QSP meet this goal of predicting clinical cytokine dynamics for my assets of interest. And this is tebentafusp. I'll be talking a little bit more about what tebentafusp is in the next slide. But this is one of the CD3-based bispecifics, and I believe this is the only CD3-based bispecific that's currently approved for solid tumors. And they do have clinical data that was published both on what cytokine dynamics looked like clinically over time, as well as they've published other information that's useful, such as PopPK models, other preclinical in vitro data, etc., that's useful for our purposes. And so the modeling workflow that was taken to try and meet this goal of predicting cytokine dynamics clinically for my bispecifics of interest was to use and leverage this published clinical data from tebentafusp to basically build and calibrate a model to the tebentafusp data, then to translate it to my bispecifics of interest, which would be done through updating different parameters related to dosing, PK, etc. And from there, I've now got a model that's at least sort of clinically calibrated. I have some clinical confidence in that I can start using to predict dose responses of my compounds of interest. And from there, once clinical data starts to become available, I can do my sort of typical modeling, validation, going back and updating with the clinical data as it becomes available. So this was the modeling work stream that was performed. Again, for the purposes of today, I'm not going to be able to talk about any assets internally that we're working on. So I'm going to focus on kind of this first bucket of developing and calibrating the model to this publicly available data from tebentafusp. So just briefly a little bit about tebentafusp. Again, like I mentioned, this is a FDA-approved CD3-based bispecific. It was approved a little over a year ago for unresectable or metastatic uveal melanoma. So this is a melanoma of the eye. Again, I believe it is currently the only CD3 bispecific that's approved for solid tumors. And as I mentioned, I'm particularly interested in this compound and the data, clinical data from this, because it provides this analogous clinical data that I can kind of leverage to help inform what I might expect some internal bispecifics to look like. So again, I have not personally worked on tebentafusp, but my understanding of how this compound works, it follows pretty typical MOAs that we'd expect from these T-cell engaging bispecifics, where it binds to CD3 on the T cells, and it binds to this, what I'm going to call GP100 antigen on our uveal melanoma cells, really resulting in targeted T-cell killing of these melanoma cells, cytokine release, everything like that that follows. So with that, here is the QSP model that was developed to capture the tebentafusp clinical cytokine dynamics. I'm not going to go through everything in super great of detail. Again, I think Rebecca mentioned at the beginning, there was a paper I published on this at the end of last year, and I'll flash that up at the end of the webinar. So if you're interested in, sort of the nitty-gritty technical details of this model, I'll direct you over to that publication. But just to highlight some of the key points of this model, basically, it really doesn't look any different from typical QSP bispecific models that you might have seen before, other than that there's this immune cell component I'll touch on in a second. So what we've got happening in this model is there's basically a two-compartment model to capture the PK of tebentafusp, and then tebentafusp undergoes disposition both to the tumor, so our uveal melanoma cells, as well as to skin. So skin was added as the organ of interest for our on-target off-tumor toxicity, because this GP100 antigen expressed on melanoma cells that tebentafusp targets also has expression on healthy melanocytes. And so the idea was, in order to make sure that I'm sort of capturing our safety signal, our cytokine release, our CRS potential that might be occurring clinically, I wanted to include both the on-target on-tumor as well as the on-target off-tumor effects. But once tebentafusp enters the tumor or the skin, it undergoes our kind of typical binding patterns that you've probably seen before in other bispecific binding models, where it binds CD3, GP100 to form this trimeric complex. And then what's happening over here on the right is kind of what's new that you might not have seen before. So this is a way of capturing the immune cell dynamics and the cytokine release that results from this trimeric complex forming. So I'm essentially assuming that there are immune cells. These are generalized immune cells. These are not specific, NK cells, dendritic cells, whatever. This is basically a generalized immune cell pool. And these immune cells are assumed to exist in one of three states: this non-secreting sort of basal homeostatic state, a secreting state, or a refractory state. So like I mentioned, this non-secreting state, these are where the cells are sort of in their basal homeostasis. Once this trimer formation occurs, it's going to trigger the immune cells to enter the secreting state, where they are going to be actively secreting cytokines. And after a period of time, they're going to enter this refractory state, where they can no longer be triggered by the trimer formation to release cytokines. And they'll be in this refractory state for a long time before they kind of enter back into our basal homeostatic non-secreting state. So again, that's the QSP model. I'll again direct you to the publication to get some more details about the specifics, but hopefully that gives you enough of an idea of what we're looking at, that we can start talking about, using this model. So the first step was, again, just to calibrate the model to the clinical tebentafusp cytokine dynamics that have been published. So these are the 5 different cytokines that were publicly available from that publication. There was IFN gamma, IL-6, IL-10, CXCL10, and CXCL11. So these plots, and most of the plots that I'll be showing from here on out that are actually, simulations from the model, are going to be shown in this way, where it is time on the x-axis and the y-axis will be the fold change from baseline of the cytokine. I don't think I mentioned this drug is given once weekly, so this is across 8 doses of this drug. And I know that you can't really see the information in the slide super well, but the slide is just to show that this model was able to be successfully calibrated to these tebentafusp clinical cytokine dynamics. Again, I have more information about some quantitative measures of goodness of fit in the publication you can look at if you're interested. But this was just to ensure that the model could be calibrated. And I would like to kind of also point out, whenever we're talking about model development and QSP modeling, there's kind of two steps, right? There's model calibration and then model validation. So I really want to emphasize that this model has been calibrated. It has not been validated in the context that I'm showing you today. So again, like I mentioned, I have not worked on tebentafusp. I don't have access to individual patient level data from this compound. I only have access to what is publicly available. So this model has been calibrated to what is clinically publicly available, but really not validated since I don't have a validation data set that I can use. So I just like to throw that out there just to, caveat that with everyone so we're aware of where we're standing. So with that, we can start to use the model to try and understand what's going on with our bispecifics and to try and wrap our head around how can we start thinking about dosing these bispecifics. And so this slide, again, there's a lot going on in this slide. I'm not going to walk through all of the details. Again, I'll orient you to the publication, which I'll flash at the end of the webinar, but all these figures are pulled directly from that publication. But kind of the first step, whenever building these types of models, especially I think these QSP models where we don't necessarily have data to validate against, I think it's good to do sort of sanity checks to make sure that your model performs as kind of expected. And this is a little bit of a sanity check to make sure our model performs as expected. So using the model, I was predicting where is the main driver of our cytokines coming from. And the model really predicts that this on-target off-tumor effects is going to be the primary driver of cytokines. And so that means that our on-target off-tumor effects are the main driver of toxicity. And I think that, at this point we have pretty good understanding of bispecifics that we've seen over multiple bispecifics. It really is those on-target off-tumor effects that drive our toxicity. So this is showing that, the model is basically predicting kind of what we would expect to see based on what we know clinically occurs. Okay, and from there, we'll get into some more model applications. And this is where I'll spend a little bit more time kind of walking through ways that we can use this model to help us think about dose selection for these bispecifics. So one thing I touched on earlier is this idea of step-up dosing for our bispecifics. Well, I think one challenge when we're thinking about step-up dosing is how many step-ups should we be giving? Should we be doing step-up dosing at all? Should we just be, getting flat dosing of our drug? Should we do one step-up? Should we do two? Should we do 3, 4, 5, 6? Theoretically, the more step-ups we give, the safer the drug should be. But of course, we have to balance this with practicality. To try and give, I think 5 step-ups practically is going to be an extreme challenge because now you've got to basically make 5 different formulations or 5 different concentrations of your drug that the patients are going to have to get over time, and just the logistics of that are going to be extremely complicated. So we can use this model to help us understand how many step-ups is optimal for making sure our drug is safe. And so again, in these slides, there's a lot of information that I'm not going to necessarily cover in great detail. I'll try and cover the main points. But this is a simulation that was looking at CXCL10 fold change from baseline over time. CXCL10 was kind of just chosen as representative of the different cytokines that are in this model, and looking at stepping up from 0, so flat dosing, up to 5 step-ups, and looking at what is the peak CXCL10 fold change from baseline that we see across these different dose regimens. And on the left, this table shows what those different dose regimens are. So 68 micrograms once weekly was the target efficacious dose of tebentafusp. So these are just different ways of stepping up to that 68 microgram QW dose using 1, 2, 3, 4, or 5 step-ups. And over here on the right, I'm showing what is the peak CXCL10 fold change from baseline that was achieved with some confidence interval in there. So for example, if you give 0 step-ups, you have a peak CXCL10 fold change from baseline of about 72. Once you go up to one step up, using 30 micrograms up to 68, you get 55 peak fold change, etc. And so what this model application kind of shows is that, well, once we get up to 2 step-ups, we're basically not really achieving too much added benefit in terms of CXCL10 Cmax reduction beyond 2 step-ups. My criteria for this was basically once we no longer see, I think it was 10% or 20%, I forget which one it was, reduction, that's where I'm saying that we're no longer seeing a benefit in an added step-up. So a nice way that you can kind of use these model applications for your compound of interest to help figure out, do we need step-up dosing? How much step-up should we do to get a safety benefit? So that previous slide was really just looking at different step-ups using sort of a 2-fold change with each step-up. This simulation is a similar concept, but rather than saying we're going to do 2-fold step-ups with each step-up, so 2-fold increase in dose, let's try now a scenario where we want to play around with ranges of step-up dosing. So in this case, I have simulations where I'm doing 3 step-up doses up to that target efficacious dose of 68 micrograms once weekly. And I'm going to try out some different scenarios where the lowest dose I'm going to give, the first step up, will be 10. So now I've got a dose range going from 10 to 68. Now I'll try a simulation where I narrow that dose range a little bit. So starting at 20 up to 68, and so on and so forth, up to step ups that have a very narrow dose range, only 56 up to 68. And we can play around with these kinds of simulations to look at what are our fold change from baseline of the cytokines look like to help figure out how wide of a dose range can we go and what might that look like. One other interesting thing that this model was predicting is that based on the range of the doses that are applied during the step-ups, you might actually see differences in Tmax. So the actual day at which your maximum fold change from baseline occurs might differ based on the range of doses that you apply. So for example, when I start with the widest dose range of 10 up to 68 in purple, our Tmax occurred at day 22, whereas at the narrowest dose range from 56 to 68, our Tmax occurred at day 1. And so again, I think that this is some interesting information that you could obtain from a model like this to help figure out, based on different dose regimens we're giving, when are we expecting CRS to most likely occur? Is it going to most likely occur after the first dose? Or is it most likely going to occur after a second dose, a third dose? And that might help you start to think about, when do we need to monitor these patients a little more closely? When do we need to be careful with hospitalization? Okay, for the sake of time, I'm going to go through these next 2 slides very quickly. Again, there's a lot of information in them, but I'll try to cover the key points. So one, concern that was potentially raised is that as we're thinking about doing these step-up dose regimens, is this going to affect our efficacy of the bispecific? Because you can think about, we have our target efficacious dose, so in tebentafusp's case, 68 micrograms once weekly. Well, if we're doing multiple step-ups to get to that target efficacious dose, we're going to be in a scenario where we are potentially giving our patients subtherapeutic doses for a period of time, for weeks, for months, depending on our dose regimen. And so one way that we can use this model to explore this idea of, is step-up dosing going to affect our efficacy, is by looking at the trimer formation within the tumor. And so this was a simulation that was looking at, again, those step-up dose regimens from flat dosing, 0 step-ups, up to 5 step-ups, and looking at that trimeric complex formation in tumor. And what the simulation showed is that while we do expect there to be a little bit difference in terms of the absolute number of trimers that are formed over time across step-up dosing, the actual amount is not that different between flat dosing and step-up dosing. It was less than kind of 20% difference in the amount of trimeric complexes that are formed over time. And so this model, I think, gives some confidence that we can apply step-up dosing without it expecting to affect our efficacy. And then the last sort of simulation application that I'll cover, this is just sort of a traditional sensitivity analysis. So looking at a couple of different model parameters, doing some fold change from baseline of that parameter value and seeing how it affects the CXCL10C max that's released. So again, I'm not going to go through this in super great of detail, but I think that, you know, whenever we have these types of models, particularly QSP models, this is a really nice additional application of just helping us understand our system, especially when we start to think about, you know, maybe using this model to design future bispecifics that are going to be coming down the pipeline. You know, we can take this model to some of our drug discovery colleagues and we can say, you know, based on what we've seen with changing clearance of our drug, with changing affinities to targets, target expression, things like that, you know, this is how we expect the safety and the efficacy of this compound to sort of change with these different potentially controllable parameters. And so this will help us figure out, you know, maybe how we want to design our next bispecific. Maybe it will help us figure out how we want to select a patient population, a tumor population to try and target with different bispecifics that we've got maybe earlier in the pipeline. And so with that, I'll end with just some brief future directions. You know, I think whenever we're doing modeling, there's always kind of potential for next steps and, you know, where do we go next with these models? And I think one future direction that could be done with this model is to extend the model scope. So like I mentioned, I'm really not modeling any specific cell types within this model. It's sort of a generalized immune cell pool. I think expanding it out to maybe specific cell types could be useful for helping us better understand how cytokine release syndrome occurs. The model currently does not either include tumor growth inhibition. So that's definitely something that could be of interest to include. And, you know, again, the whole goal of this model is to try and mitigate CRS in our clinical studies. As of right now, I'm kind of looking at CRS by using cytokine Cmaxes as an analog. I think there's some interesting work that could be done to maybe link some of these cytokine Cmaxes through maybe probability models, logistic models, to help figure out, you know, if we see cytokines of this level, what does that mean in terms of really CRS potential? Expanding the model to include actual CRS management. So another thing that's being done with bispecific therapies currently is, you know, maybe pre-dosing our patients with something like tocilizumab to try and mitigate that CRS that might pop up. So this model could be used to explore some of those ideas. And then there's the idea of applying this to other bispecifics. There's currently, I believe, over 50 CD3-based bispecifics that are in clinical development, and then there's all of these non-CD3-based bispecifics, such as CD137, dual targeting tumor-associated antigens. You know, I think that this model could potentially be a nice platform model to try to help explore some other bispecifics also. And so with that, you know, brief summary, this model was used to help provide some mechanistic insight into the cytokine attenuation we see through immune cell desensitization, the importance of the on-target off-tumor effects, as well as helping us understand safety in terms of cytokine release and efficacy in terms of tumor trimer formation across step-up dose regimens. And like I mentioned, this work was published somewhat recently in CPT PSP. So if you're interested in learning more, take a look there, and please feel free to reach out to me if you're interested. And with that, I'll just thank, you know, all my colleagues at Astellas that helped contribute or helped me with this work in some way or another, and happy to spend the last couple minutes taking any questions. Thanks for your attention.
Thank you, Jared. We do have some questions. And we do have some of them are duplicates, so I am going to sort of combine them together. So can you talk a bit more about what is the mechanistic understanding between less CRS with the step-up dosing?
Sure. So let me come back to kind of model schematic. So the mechanistic understanding is basically my understanding, and I believe that this is kind of the current hypothesis of the field, and the way that the model captured it is that as you start administering your bispecific, you're going to be transitioning immune cells from sort of this basal state where they have the potential to secrete cytokines to a refractory state. So you're sort of exhausting your immune system from being able to release cytokines. So the idea with the step-up dosing is by giving a lower dose, you're exhausting some of your immune cells. So you're reducing that available pool of immune cells to release cytokines. And then as you step up, now there's less immune cells available that you're going to start seeing a little bit less activation, a little bit less activation. And that kind of differential between, sort of a flat dose and step-up dosing is what results in this reduction in cytokine Cmax. Hopefully that made sense. Hopefully that answered your question.
Yes, and it did lead to the second question in this series, which is, can you use this model to optimize both the step-up dosing and the timing of the dose?
Yes, I believe you can. So everything that I'm showing in this webinar and also in the publication was just based on tebentafusp. So I only looked at and showed once weekly dosing, but you definitely could, change the dose regimen in any way you want. You could give dose interruptions. You could give, biweekly dosing. You could give, switch from weekly to biweekly. Yeah, really anything you want.
Okay. You mentioned how your QSP model is similar to other models of bispecific interactions. With the current understanding of the underlying mechanisms and available data, do you see the possibility or value of developing a more generic model that could be used to study a wider bispecific compound, a wider range of compounds and cancer types?
Sure. Yeah, that's a good question. And yeah, I guess just to clarify, I was kind of saying that if you look at, there's kind of two different pieces that are happening within this model. So there's the binding of the bispecific to form the trimer, and then there's what's happening with the immune cells. So that first piece, the binding of the bispecific to form trimers, that's what I was referring to as similar to other models out there. I do think there is the potential to kind of make this a little bit more generic so that it could be more readily applicable across bispecifics. One way of doing that would be, again, since I was looking at tebentafusp explicitly, I have this skin compartment to account for the on-target off-tumor effects. I think this could be replaced with kind of a more generic healthy tissue compartment to account for on-target off-tumor effects more generically. So that's something that could be done to make it, potentially more readily applicable across bispecific types.
Do you have actual numbers of immune cells in the model, or are they modulated on a range of 0 to 1, so a normalized cell number?
There are actual numbers of immune cells in the model. That's all in the supplemental material of the publication, so you can take a look. But those are basically calculated by, accounting for the tumor size and the skin cell size and what the cell densities in those were. So yeah, I think all the calculations are in the publication.
So you noted that the model was predicting cytokine kinetics. Could you add to this model and predict actual CRS events?
Yes, so sort of briefly touched on that in next steps. So I think what you would have to do for something like that is have some way of linking cytokine concentrations with CRS events, which at the time that I was working on this model, I don't have the data to do that. So again, I didn't work on this compound. I don't have patient level data, but theoretically, you could take the patient level data. You could look at, this patient had an X-fold change from baseline in, IL-6 or whatever, and you could use sort of a probabilistic or logistic model to do basically an exposure response of cytokine exposure versus CRS incidence response. And now you'll basically have this QSP model linked to a probabilistic model to say, if this cytokine is above a certain threshold or within a certain range, that's going to lead to this potential of CRS incidence. So yes, definitely possible, something I'm very interested in, but quite frankly, don't have the data to do quite yet.
With that in mind, one of the other questions, actually a couple of the other questions that we have is, could you add in the treatments for CRS and see if you can dose and time both types of drugs to reduce the
Yep,
CRS?
definitely. You could do that. So yeah, you'd basically just have to add in, something like tocilizumab. You can build in, the PK for that. You can build in its disposition to the tumor, skin, or healthy tissue, how it interacts with IL-6 to, try and provide a holistic representation of, timing of dosing for tocilizumab, dose of tocilizumab, what that would look like in terms of cytokine dynamics and CRS potential. Yep, another thing I'm very interested in.
And could you briefly mention how this model for a solid tumor might be different than models for a non-solid tumor?
Sure. Yeah, so I think really the key difference would probably be what our sort of MOA assumption is. So I think, my understanding of the current hypothesis of the field is that when we're talking about cytokine release and CRS in, for example, hematology, liquid tumors, it's not so much actually, the immune cells that are driving cytokine release and CRS. It is actually more driven by this trimeric formation between your tumor and your T cells. So it's driven by, like with blinatumomab, I think they saw a very nice correlation between CRS with B cell reduction, your tumor B cell reduction. And so the hypothesis is that your CRS for hematology is actually more driven directly by your on-target on-tumor effects. And the hypothesis for solid tumors is it's more driven by the on-target off-tumor effects. So it might be a little bit more important for the solid tumors to account for what's going on in terms of immune cell dynamics.
Okay, I'm going to mention right now to our audience that we have a lot of questions, and I know that our speaker is not going to be able to address all of them in the time we have left. So we will be sending these questions on to our speaker after the webinar, just to give everybody a heads up. So there are a lot of questions about the cytokine production and the immune cells and how you captured that data.
Sure.
And in particular, feedback maybe between the cytokines and the immune cells.
Yeah,
So,
it's a great question. So, I mean, the simple answer and the honest answer is I think this is a relatively simplistic way of capturing the cytokine release currently. So there's no feedback from the cytokines to immune cell dynamics. And like I mentioned, these are generalized immune cells. These are not specific immune cells. I mean, I would kind of argue that if you're looking at just this immune cell piece by itself, this is sort of a more like empirical way to capture the cytokine dynamics that we've seen clinically, which is why I mentioned, for next steps, I think the first thing that I'd be interested in is sort of expanding this model scope, figuring out what are the actual cell types that are being triggered by our trimer formation, what are the actual dynamics of these different cell types, what are those feedback or feed forward mechanisms, and how do we include them? I mean, quite frankly, I think a lot of that is sort of unknown in the field, and that was why this sort of more generic approach was taken as a way that we can still capture and represent the clinical data and still use it to make clinical decisions, even if we don't necessarily know all of those granular level details. But I'm definitely interested in expanding out this immune cell component to try to better tease apart and better represent really what's happening with the immune cell, because I recognize that that could be important for different kinds of bispecific therapies.
Thank you, Jared. We are out of time, and again, we will be passing these questions on to our speaker. Thank you for a wonderful webinar. This was really interesting, and this is really a hot topic in many areas, is handling the toxicity versus the efficacy for different drugs. So it's great to see this applied in a QSP model and looking at both aspects. So I'd like to say really thank you for the presentation, and we'll hope to see everybody back again at a future webinar. Thank you, Jared.
Yep, thanks for letting me share. Thanks for your attention, everyone.
Thank you, everyone. Have a great day and a great month. Bye.